Hello everyone. So this is practical 2 by our groups. The title of this experiment is Spectrophotometry. First and foremost, let me introduce my group member, which is Toyi, Dover, Armau and Manjay. And this is our picture. The spectrophotometry experiment aims to familiarize students with the qualitative and quantitative analysis of biological samples and molecules using spectrophotometry. Laboratory skills to be acquired include determining the absorbance spectrum of unknown compounds for qualitative analysis and preparing and graphing standard or calibration curves for quantitative analysis using colorimetry. Practical methods involve preparing test solutions, measuring absorbance, plotting calibration curves, and estimating unknown protein concentrations using provided data and standard curves. Through hands-on experimentation, students develop essential skills in spectroscopic analysis for biochemical research.
The materials that we use is spectrophotometer, cuvette, Bradford reagent, distilled water, test tubes and test tube racks, micropipetters, 70% ethanol solution, 1 mg/ml BSA protein, and unknown protein solutions labeled A, B and C.
Now methodology, part 1. Place 4 milliliters of distilled water in 7 test tubes. Use micropipetters. You can see my friend's hand shaking just to make sure the volume we pipette precise. Next, place 1 milliliter of Bradford reagent into each tubes. We are sorry because we forget to record video when we place the reagent. Anyway, as you can see, the clear water turns into a bit purple when the reagent placed. And then add in 50 microliters, 100 microliters and 200 microliters of BSA protein standard. What prepare in duplicate mean? I will explain with the picture. So there are 7 test tubes, right? The first 2 test tube, we add 50 microliters, the second 2 test tube, we add 100 microliters, and the third 2 test tube, we add 200 microliters of BSA protein standard. How about the last test tube? Don't add anything, let it only water and reagent because it will be our 0. Next, prepare for reading the sample. Just transfer each of the solution to cuvette. Read the samples from least concentrated and record the absorbance for each. Use sample without protein as blank to zero the machine. And you can watch the video to see the spectrophotometer that handled by our lab assistant. Also as evidence, you can see my friend jot down the absorbance. Moving on to the methodology, part 2. It almost similar to part 1, so let me reverse first. So same as part 1, place 4 milliliters of distilled water in 7 test tubes. Use micropipetters. Next, place 1 milliliter of Bradford reagent into each tubes, also same like part 1. And here are the difference. Add 100 microliters of sample A, B and C like the first part, which is prepare in duplicate. And also don't forget the blank or 0. Then, similar like part 1, prepare for reading the sample, just transfer each of the solution to cuvette. And last, read the sample and record the absorbance that's shown on the spectrophotometer.
After conducting the spectrophotometry experiment and analyzing the results, it is evident that students have gained valuable insights and practical skills in the qualitative and quantitative analysis of biological samples and molecules using spectrophotometry techniques. By accurately determining absorbance spectra of unknown compounds and preparing calibration curves for protein concentration determination, students have demonstrated proficiency in laboratory techniques essential for biochemical research. And lastly, this is my group reflection through this experiment. That's all, thanks.